This is Dr. Vishnu Sankar Sinha, PG Department of Botany, Tata College, Chaivasa, Kolan University. Now, today I am going to discuss mutation. The term mutation was proposed by Hugo de Veris while he was working on Oenothera lamarcania. It is an autotetraploid. But the phenomenon of mutation was first noted in 1791 by Seth Wright, noticed short-legged lamb in the flock of Ascon sheep in England. Now, mutation has been defined by the several geneticians or other scientists. Darwin called mutation as spots for sudden appearance of new hereditary character in the offspring. Watson defined mutation as discontinuous variation. Stebbing defined mutation as discontinuous chromosomal changes with genetic effect. According to Hugo de Veris, mutation is a sudden and drastic heritable changes not traceable to segregation or recombination. And last, for Wisher, in 1968, mutation is sudden change in heritable character due to an alternation of their number, molecular structure, or sequence of nucleotides. On the basis of cell, mutation may be of two types. One is germinal mutation, another is somatic mutation. Germinal mutation occur in germ cells, that is zygote, gametes, spore, etc. Germinal mutation passes from generation to generation and it is stabilized in population, while somatic mutation occur in somatic cells and it is not passes from generation to generation, not stabilized in the population, and it is disappear with the death of affected individual. Some characteristics feature of mutation. Number 1, mutation are random. They may occur in any gene at any number of times. Number 2, mutation are generally harmful, only 0.1% useful. Mutations are generally recessive, but dominant mutation also occur. Recessive mutation express itself only in homozygous or haploid condition, while dominant mutation may express in homozygous, heterozygous or haploid condition. Role of mutation. The role of mutation is ultimate source of all genetic variation and it provides the raw material for evolution. Number 2, mutation leads in the genetic variation with the mixing of alleles. Mutation provide a raw material for the evolve and adapt to different environmental condition or they change according to the environmental condition. Now next is the rate of mutation. The rate of mutation is observed in case of codon, in case of nitrogenous base pair or locus. The rate of change in codon is 3 into 10 to the power minus 8 to 3 into 10 to the power minus 9. Rate of change in nitrogenous base pair, that is 10 to the power minus 8 to 10 to the power minus 9, or rate of change in locus, that is 10 to the power minus 4 to 10 to the power minus 7. Types of mutation. on the basis of their action. First is forward mutation, the mutation of a wild type gene to form that result in mutant phenotype is called as forward mutation. Number 2, backward mutation, the mutation in a mutant gene which result in wild type phenotype is known as backward mutation. Third one is suppressor mutation, a mutation that partially or completely cancel the phenotype effect of another mutation. Fourth is lethal mutation, a mutation that is fatal to the organism. Fifth is silent mutation. Silent mutation is when a gene occurs which show no change in phenotypic characters. That is, when AGA codon is changed into AGG codon, here both the codon for arginine. And similarly, when GUA codon change into GUG codon, here both the codon recognizes the valine amino acids. Now, sixth one is nonsense mutation. Nonsense mutation means any codon change into termination codon. And last one is biochemical mutation. Biochemical mutation, here organism cannot synthesize an essential amino acid due to lack of one particular enzyme lead to change in the phenotype of an organism. This picture clearly show that the missense mutation, silent mutation and nonsense mutation. The codon UCU for serine, when U is changed by A, then UCA codon also recognize serine. This is called silent mutation. Similarly, when UCA change into ACA, the UCA for serine while ACA is for threonine, and this is missense mutation. Or when UCA is changed into UAA, the UAA is termination codon and they cannot recognize any amino acid, that is known as nonsense mutation. Gall 1964 mutation is classified into two broad categories. One is macro mutation, another is micro mutation. Macro mutation is also known as chromosomal mutation or false mutation. Macro mutation is of three types. First is chromosomal aberration, aneuploidy and polyploidy. In chromosomal aberration, the change in chromosome structure but chromosome set and number remain constant, while in aneuploidy, here only change in chromosome number due to addition or deletion of one or more chromosome number, but here the chromosome set remain unchanged. And in case of chromosomal polyploidy, here change in the chromosome set. And second is micromutation or gene mutation or point mutation or true mutation. It is of three types. First is frame shift mutation, another is transition mutation and transversion mutation. Frame shift mutation is due to addition or deletion or rearrangement of the nucleotides, nitrogenous bases of DNA. Transition mutation, here purine is changed by another purine or pyrimidine is changed by another pyrimidine. And in case of the transversion mutation, the purine is changed into pyrimidine or pyrimidine changed into purine. For detail, no, this one not, no. Yes. We know the four nitrogenous bases, that is adenine, guanine, cytosine and thymine. The adenine and guanines are purine and cytosines and thymines are pyrimidines. Here purine AG or pyrimidine CT is interconversible. This is known as transition and transversal mutation. Now due to the frame, in case of frame shift, When a DNA segment is AUC, AUC, GUC, now A is addition, then the codon will be AAU, CAU, CGU. Now this is a totally different DNA fragment which recognizes the different amino acid. When in deletion of A, then AUC is changed into UCA, then UCG and UG. It is the another fragment of DNA. Now the next is the gene mutation. Now the gene mutation is of two types. One is a spontaneous mutation, another is induced mutation. A spontaneous mutation occurring in nature without any treatment in a population occurs due to unusual base pairing and occur during DNA replication. DNA fragment either lost or change their structure. While induced mutation caused by treatment of physical or chemical mutagens or agents. Mutagens may be of two types, that is physical mutagen or chemical mutagen. Physical mutagens may be non-ionizing, that is ultraviolet, or ionizing, that is electromagnetic rays, X-rays with 0.10 to 10 nm, gamma rays 10 to the power minus 4 to 10 to the power minus 1 nm, or corpuscular rays, that is beta ray, protons, neutrons, alpha rays and other heavy particles. Similarly, chemical mutagens may be alkylating agents, that is sulfur mustard, nitrogen mustards, ethyl methane sulfonate, ethyl ethane sulfonate, diethyl sulfonate, etc. And is acridine dyes, that is acryflavine, proflavine, erkidine orange, erkidine yellow, ethidium bromide. Third is aziradine EI, ethylene imine. Next is base analogues, 5-bromouracil, 5-chlorouracil, 2-amino. Now fifth is deaminating agents, that is nitrous acid HNO2 and others. Malic hydrazide MH, sodium azide SA, hydrazine and hydroxyelamine. Now HNO2 removal of amino group from adenine and cytosine, converting them to hypoxanthine and uracil respectively, ultimately leading to transition mutation. Now in tautomeric shift, the keto form is converted into enol form, or amino form is converted into imino form. Amino form is common and imino form is very rare, and similarly in case of the adenine, amino form is also converted into imino form, or in case of also in guanine, the keto form is converted into enol form. By the action of EMS, the guanine is changed into 7-methyl guanosine. At the 7th position, the ethyl group is added in the guanine and leading to the transition or transversion mutation. Now the application of mutation. Mutation causes genetic variation by inducing recombination in linked gene, lead to new varieties. Number 2, in the field of agriculture, it induces qualitative and quantitative characters improvement. Number 3, mutation causes induction of desirable characters. Number 4, in pharmaceutical industries, it either increases the rate of reproduction in microorganisms or secondary metabolites or active components of plant increases. Number 5, mutagenesis help in deciphering the hidden messages of DNA and thus genetical material. Some common mutant variety are as available due to the mutagen, that is Sharvati, Sonara, NP806 of wheat variety due to the gamma ray mutation, while Jagannath is due to the gamma ray mutation of rice, and in sugarcane CO 8152 it is also due to gamma ray, and MCU of cotton due to also of gamma ray. The in case of the groundnut, TG1 and TG19 is also produced due to the gamma ray induction. Now, the demerits of mutations. The demerits of mutation is it is very low frequency of desired trait, mostly unfavorable results, highly unpredictability, and requirement of a large test population, and number 5 generally they are recessive in nature, and number 6 generally they are related with linked gene, thus causes undesirable changes in the organism or crop. This is overall the gene mutation. For further you may consult the book of Dr. P.K. Gupta or Stebbins or Watson. Thanks for watching.